Hey everyone, and welcome back to another episode of Snakes on the Plane. Today we're going to be talking about genetics and really kind of a deeper than skin deep sort of conversation. I didn't really want to just buzz over the logistics and say, okay, now you know the basics. Because when I was starting, I really was craving more information, and I am not going to go too crazy into it, but I feel like if we understand how the genes work and where they're at in the DNA and what they cause and so on and so forth, and what else is available just besides the three main genetics in ball pythons that most people know, which is incomplete dominant, recessive, and dominant. There's more than that. So we're going to be going into all those today, and I hope you guys are just as excited as I am about it. So first, before that, I do want to apologize for not getting videos out there as often as I would want. I've been having a crazy amount of schoolwork as I'm still in college. The holidays just hit, and I've been doing all this, not all this, meaning taking care of the animals and the house and everything while my partner is out for a little bit with some training in another state. So I will be trying to get things out quicker and more videos out for you guys. Please let me know what videos you would like to see. And without further ado, here we go. So what's going to show on your screen right here in a second is a hurricane ball python, and we're going to start off with the kind of gene it has. It has an incomplete dominant. Now you might be thinking, oh, I've heard of codom before, but not incomplete dominant. Codom and incomplete dominant are supposed to be or mean the same thing. However, technically their definitions are really different. So incomplete dominant is the correct one. Codom would actually mean like a third of the snake might be, if the snake was a combo, let's say it was a pastel hurricane, a third of the snake would be pastel, then the next third would be hurricane, and the next third would be pastel. It'd have like segments of the genes, while incomplete dominant is sort of a mixture, but it's a little bit more complicated than that, and we'll get into that right now. So what I'm going to show you guys is a Punnett Square, and that's this right here. And basically, this just allows you to visualize what's going on with the genes from the dad or the sire and the mom or the dame. You can find calculators that just tell you the odds anyways, on like World of Ball Pythons and Morph Market, and I'll put a link in the description to those. But it doesn't give you a Punnett Square. So for those of us who are visual learners, it does not show you exactly what's going on, which I find to be helpful. And so let's get started on that. So if this snake right here is a hurricane, say the dad is, he's going to have an H gene for hurricane and then an N gene for normal. And so why does he have this gene? Well, what is the normal gene? It just basically means there is a normal gene, or the gene that's normally there, with the hurricane gene, because you know the mom and the dad obviously give one copy of their two genes to their offspring, and these four squares right here represent the offspring. Awesome. And so if the mom is just a normal, then obviously she's just going to have the normal gene she normally would at those normal spots. It's really kind of makes me feel bad for saying it like that and kind of just teaching it dumbed down, but that's basically all it is just the normal gene at the normal spots. And people are like, well, what is the normal gene? Just the one that's normally there. It might be a different gene at different places, but it's just the one that's normally there for snakes. So if we start combining these, kind of like a times table in math, what we get is you go up first and then you go left always, and boom, that's the first one. So I'm going to fill this out and we'll be right back. Alright, and this is what you get. So half of the offspring that we can see right now are going to be the same, and half of them are going to be the same over here. So we can obviously see right off the bat that there is a 50-50 chance of getting these two different kinds of phenotypes, and phenotype means different kinds of looking snakes or morphs from one egg. So boom, already super exciting. 50-50 chances is pretty good. So right here we see that the dad passed on his hurricane gene and the mom only has normal, so she can only pass on that normal gene. So just like the dad, same genetics. So this snake right here is a hurricane, and so is this one. And these two are normals. So that's awesome. So what we're going to do now is pretend that it's been a few years, and we'll say that this snake right here was a female, and we grew her up, and now we're going to breed her back to her dad. And for those of you that are kind of newer to this, that might sound really weird, but inbreeding in reptiles is much different than inbreeding in mammals. Some people say you should go about like 4 to 5 generations and then get new blood in before trying to inbreed again. Some people even say 10 to 11. So that needs more research done if you ask me, but you can definitely breed back parents to sibling, or parents, siblings, and parents to offspring without any trouble for a couple generations. So I'm just going to get this Punnett Square remade for you guys. Alright, so now that we're back, we can see that this gene right here has two of the same genes or traits from 2 different parents. This one and this one may look different. It really doesn't matter if these two are mixed up or not. You can write it either one of these 2 ways. It does not change the genetics of the snake at all. It just is because this N was up top, so you always take the N straight down, and then you go left to right. So these 2 are actually the same, so it's a 50% chance for these traits, and then a 25% chance for either one of these traits. And so what does the 2 copies of the same gene mean? Well, that becomes a super hurricane. And I'll put a picture up for you guys right now. You can really see a difference between this super hurricane and this single gene hurricane that both the mom and the dad were. If you take a look back at the super hurricane, you can see that all the alien heads are really blown out and they just swirl nicely. It kind of reminds people of GHI, except for the pattern is a lot more tidy, and also the dorsal is quite dark with a reduced dorsal stripe. Now, it does, or super hurricanes do seem to be, and hurricanes in general seem to be more of a chocolatey color than GHI and other mutations like Trick and Blitz. Trick and Blitz are also dominant, so that's a difference between them and hurricane. And then if we go back to the single gene hurricane, you can really see that those alien heads haven't been blown out completely, and that there just kind of is more madness going on in the single gene versus this super hurricane. And the super hurricane is actually called a Hayabusa, and these photos both are courtesy of Hans Wenner in Germany, the man who founded the morph. So if you guys want to see more hurricane combos, go check out his page on Facebook. There's plenty there to gawk at. Alright, now getting back to the Punnett Square. So you can see that there is a difference in what the snake looks like depending on if it has 1 copy of the hurricane gene or 2 copies of the hurricane gene. And that is what makes it co-dominant. We can see a difference in the super form, which is also called the homozygous form, versus the single gene form, which is also called the heterozygous form. So homozygous means same, and heterozygous means different. Basically, in layman's terms, it's that easy. So if they have the same gene next to each other, they're homozygous. And if they have a different gene next to them, they're heterozygous. And this is going to come into play more later, but I just thought I'd throw that out there so that we can repeat it a few times and get it in our brains. So if the genes are the same, like hurricane, both of these genes are the same, then they should go to the same place on the snake. And what I mean by that, or like on the DNA, what I mean by that are allelic combos and complexes. And I know I just threw a bunch of words at you you may not know. And so basically, allelic genes are ones that occur in the same place or next to each other. And so obviously, if they're the same gene, they're going to occur in the same place, right? they're not going to be in two different places in the snake or two different places in the DNA. It wouldn't make sense to have a hurricane gene up here on like the helix and then another hurricane gene down here on the helix. Wouldn't make sense at all. So that's going to come into play in just a second here after we get done with our next Punnett Square. So what we're going to do now is pretend that this snake right here was a female. We grew her up in a few years and we're going to breed her back to her dad. Alright, so what we have done is bred this female back to her father, and if you look, we have a 50-50 chance right off the bat before we even have to delve into what the genetics are. We can see that. And so we can see that this is a super hurricane, this is a super hurricane, or Hayabusa, this is a normal hurricane, or a single gene hurricane, and this is a single gene hurricane. So everything has hurricane in it. And if you look over at the mother, she only has hurricane being a Hayabusa. She can't throw this normal gene. So what that means is everything that she's ever going to produce in her entire life will be hurricane, and that is what I love so much about super forms. Super pastels, super spot noses, anything like that. They can't throw normal, so everything in that clutch is guaranteed to be whatever the super form is, but the single gene version of it, of course. So, single gene hurricane, single gene mahogany, single gene pastel, super, super awesome. So if you're just starting to get into snakes and breeding, obviously do your research, but after that I would highly recommend getting a few supers in your collection because they will really kick it up a notch for you. Alright, so next what we're going to talk about are complexes. So I'm going to show you two different snakes or two different genes that are in the same complex and what that means. And these genes are also allelic. So the first one is a lesser, and as you can see, this lesser is from Breeder's Circle, as well as is the next one, which is a Mojave. And so lesser and Mojave, the genes, if you combine them, happen at the same place. They go to the same place and they interact as a superform. So what happens is you get a snake looking like this, which is a blue-eyed leucistic, or an all-white snake with blue eyes. Pretty awesome, right? But it gets even cooler. You see, these two incomplete dominant genes interact with each other on the same allele at the same place as a superform, meaning that every time that this blue-eyed leucistic snake, which is a lesser Mojave, is bred, it will only produce offspring that are either lesser or Mojave. It does not have a normal gene next to the lesser and a normal gene next to the Mojave because they occur at the same place. So I'll show you an example of that right now. So let's just say this sire is a blue-eyed leucistic, so he has a Mojave and he has a lesser gene. Now normally if these snakes were not in the same, or not these snakes, excuse me, if these genes were not on the same allele, then what you would have next to them would be a normal gene that would normally go along with it. But because these two genes are already in their superform and already taking up the two notches that exist on the DNA, then you only have these two interacting. So of course he doesn't have a normal gene, as you can see, so he only can throw that Mojave and he only can throw that lesser. So we'll just breed him to a normal female really fast, and I will be right back for you guys. Alright, and we're back. As you can see, 50% of the clutch would be Mojave single gene, and 50% would be lesser single gene. So that just goes to show you that you can have the super form of a snake with two different genes interacting on the same allele that are either allelic or in the same complex. Technically, they would be both, and these also include in the blue-eyed leucistic complex there are bamboo and Russo Mojave lesser / butter. Those two are the same genes. And then there's other complexes as well, such as fire complexes, where you can create black-eyed leucistic snakes and yellow belly complexes, and there's just a ton of them out there and more being discovered all the time. So these super forms hopefully go to show you guys why I believe that incomplete dominant is by far the coolest and obviously my favorite type of gene. And again, if you guys don't have a super in your collection and you're thinking about breeding, definitely go out and get one because it'll really, really, really help you guys out. So let's move on to dominant genes. Now, dominant genes are a little bit controversial right now. I'll go over both of the sides in a second here. And basically what you guys need to know is that when you breed a dominant gene to a dominant gene, for the sake of teaching, let's just say this pinstripe right here, which is a dominant. This photo is from Prehistoric Pets. So let's just say that you breed a pinstripe to a pinstripe. What's going to happen? Well, if it was anything like the Hurricane or the Mojave and the Lesser, you should get a super form, right? I mean, the Punnett Square would show a super form, right? Well, that's where some people say no, because when you breed a pinstripe to a pinstripe, all the babies that are pinstripe that come out of that clutch will all look basically the same. There'll be subtle differences, but they'll basically look the same. So if there was a homozygous version or a quote super version of that gene, it doesn't look any different than the parents. And so far, there's only been a handful of people that have come forward saying, hey, you know, my pinstripe snake here that came from a pinstripe pairing has only thrown pinstripe. So he must be homozygous pinstripe. He must be the super form. Which in that case, I don't believe it would be actually called a super form just because it doesn't look any different. It just probably would be called the homozygous form. So that's what I'm going to call it from now on. And then someone else, JKR Justin, said that in a YouTube video, he thought that there might be a possibility that leopard was, that he had produced a leopard that was homozygous. So there have been a couple people out there that have said so. If you go to World of Ball Pythons and other sources, they will also state that there can be a homozygous form of pinstripe, or of, not just pinstripe, of all dominant genes. And to me, that does make sense. Not only do I kind of hope for that to happen, so I might be being a little bit biased here, but it does make sense, because if you have a female and a male that can both give pinstripe, and the pinstripe happens on the same, in the same location, like we saw in the other genes, why would only one copy get passed on if they both were trying to pass on a copy? That doesn't make any sense to me. So hopefully this will all be proved out in time. I think it'd be a really exciting project. If any of you guys want to do it, it would be kind of grueling because you'd have to, you know, prove out every single offspring for several years and see if there's any, you know, discrepancies or not. But it'd be a pretty awesome project and kind of get your name out there. So why don't we just take a look at what some people believe is actually going on. So right now we got the P for pinstripe in the sire and the P for pinstripe in the dame. And then we're going to add in our ends. So right off the bat, we get just a regular pinstripe. And a good old-fashioned normal. And a pinstripe. Now this is where it gets dicey, because some people will say, yeah, you will have a homozygous pinstripe here. You won't be able to tell the difference, but you will definitely have a homozygous pinstripe. Like, it can happen. And then you'll have some people saying, well, no, like, this doesn't happen. What happens instead is, magically, it's only going to be one pinstripe gene passed on and then a normal gene there. Because there can't not be a gene next to this pinstripe gene. Like, this parent can't not put out a gene, either one of them. And so there has to be something here. So obviously it would have to be the normal gene that's next to it. See, just to me that doesn't make sense. I really think that somewhere out there has to be somebody that has made the homozygous form and then just not known it. And every time they breed, they're just like, wow, these are great odds, or whatever they're thinking. But at the same time, I don't think that anybody's that naive. So it really is just a mystery just going to throw that out there for you guys, that there will be a lot of people who are very firm in their belief that it is either the homozygous form or it is going to be the heterozygous form always. But honestly, we won't know until someone takes the time to prove it out, but nobody really wants to do that because they don't look any different, for one, and two, it's going to take a lot of time. So it'd be really cool to see some of you guys do that, and tell me what you guys think in the comments below about this. But all that you really need to know is that they don't look any different if there is a homozygous version of the gene, and the most important thing to remember is, just like in complete dominant, you only need one gene of whatever the dominant gene is, in this case pinstripe, to have a visual pinstripe or a pinstripe that looks like a pinstripe, which is different from what we're about to get into. Ah man, here we go. Good old-fashioned recessives. So right here is a recessive for you guys. This is an exanthic ball python. It is the snakekeeper line, or TSK exanthic, and this photo comes from JD. He is the one who founded the morph and has some really cool combos out there. Again, if you guys want to go to his page on Facebook and check him out, some awesome stuff. So with this snake, what can happen is kind of kooky, and so we're going to go over it a couple ways, just because I feel like it will be easier to understand being able to see it in more ways than one. So what we're going to have right here is a dad that is exanthic. So, and I'm already messing this up. So how we show recessive genes is not with capital letters like I was just doing, but with lowercase letters. And you may be saying, well, isn't this a super exanthic? Well, we'll get to that in a minute. And then a mother that is just normal, just like before. So I'm just going to fill this out for you guys, and I'll be right back. Okay, and this is what we come up with. 100% of the offspring are the same. Pretty cool, right? And so you may be thinking, well, if the dad was a sort of super form, are these going to look any different than this snake right here, which is the exanthic? And yes, they are. So with recessive genes, you have to have two copies of the same gene at the same place, or what you can think of as the super form, although it's not referred to as that. It's referred to as the homozygous form. So you have to have the homozygous form of the snake to actually be able to see a difference. So right down here with these heterozygous, or two different genes, even though one of them is exanthic, and so what you get is a snake that looks like this. Now it looks like normal, but it still has that one exanthic copy of the gene. And so what they call this is a 100% heterozygous exanthic, or just het exanthic. If something is 100% het, they're just going to call it het for short. So this is a het exanthic, or heterozygous exanthic. Sorry, I'm kind of repeating this a lot. I just find that repetition really helps people get this into their brain, because it's about to get a little bit more crazy, but not too bad. And so you can tell there is a huge difference between this heterozygous exanthic and this homozygous exanthic. If you don't have the homozygous version, the heterozygous version is just going to look like a normal. And if you do have the homozygous version, it actually looks like the gene it's supposed to be. So if we go back over to our Punnett Square, what happens if we breed our heterozygous offspring to the homozygous father? Why don't we check that out? Alright, so basically what we would be doing here is breeding this homozygous snake to this heterozygous snake, and what are we going to get? Well, right off the bat, we see that we get, there we go, a homozygous animal. So that is going to be a visual recessive, is what we call it. And then we're going to get another homozygous animal. Fantastic. And then these last two are going to be heterozygous. Awesome. So 50-50 chance of getting either a visual recessive or a heterozygous, and a non-visual recessive. So hopefully that's pretty easy for you guys to understand. I know it's a little bit different than what we were talking about. If it makes it easier for you guys to remember, you can think about it as the super form only shows the actual gene being the homozygous form. But again, nobody calls it the super form, they call it the homozygous form. But if you kind of need to lean into that for a while, you sure can. I mean, whatever works for you, right? So now let's take a look at what happens in a combo. And right here we have, on the left is a heterozygous pinstripe, and on the right is a visual pinstripe. And obviously these are for xanthic. This is also from JD, so again, if you guys are interested, please go check that out. And so how we write this up would basically be the sire being, let's just say the sire is the visual pinstripe and the dame is the non-visual xanthic pinstripe. So the sire would be pinstripe, and we just write it just like you normally would. And then we're going to write the pinstripe on the mother here. And normal. Now we'll write in the xanthic stuff right on next to it. And xanthic. And same thing for the mother, but since she is heterozygous, of course she's going to have another N here. Fantastic. So there you go. So how are we going to combine this? Well, first things first, we're going to do exactly what we did before. We go top to left, and then top to left. So top to left, and then top to left. And why that's so important is because now it's showing that these two genes are sitting next to each other, and these two genes are sitting next to each other. If you were to write it like top, then it would show that the pinstripe and the xanthic are in the same place, or like allelic, you could say, while the over here also pinstripe and xanthic are in the same place, which is obviously not true. We want the pinstripe and the pinstripe to be in the same place, since they're obviously the same gene, and the xanthic and the xanthic to be in the same place. So right here, right off the bat, awesome stuff is happening. We have that visual xanthic pinstripe. So I'm just going to fill this out for you guys and be right back. Awesome, and done. So to you guys, it may look like there is a 25% chance to create each one of these offspring, but remember that there hasn't been a homozygous dominant form confirmed yet or discovered yet. So technically, even though this is the homozygous form that we have written here, it is the same as this heterozygous form, because it won't look any different, and if it is different, it hasn't been found yet. So it is actually 50% chance on these two and a 25% chance on these two down here. So cool. Now that we have that out of the way, we have a visual pinstripe, and then a pinstripe that is heterozygous for xanthic, just like the mother here, and then just a normal heterozygous xanthic. And so what's kind of cool about this is if you have a snake that is a visual for a recessive gene, any recessive gene, if it's a homozygous or a visual for that, obviously all the offspring are going to be at least heterozygous for that, because each one of them will always get a copy of that lowercase letter, whatever it is, and a copy of that recessive gene, because the parent doesn't have anything else to pass on. So if you guys are looking to get started into recessive genes, I would highly recommend getting a visual, and then later down the road something that is het for that, because you really don't really want to waste that much time, because it can take 2 to 3 years to grow up these females, sometimes even more. And I have a male right now that's 3 years old and still doesn't really want to breed. He just would rather sit in his hide and sulk about how terrible his life is because he doesn't get a rat every night, so definitely something to think about there. So now that you guys know about recessive stuff, those are the 3 main genes that most people consider when they talk about ball pythons: the recessive, the dominant, and the incomplete dominant. However, now we're going to move on to a couple things that most people don't talk about, and I want you guys to know about. Alright, so moving on to some sort of odd and different kinds of genes that we've seen so far. We're going to start with the banana. Now, banana and coral glow came out about the same time and were thought for a while to be possibly two different kinds of genes. But as time went on, we found out they're the same thing, just like lesser and butter are the same thing. Just kind of happened to be bred out into two different lines at the same time, like I stated. So if you hear someone say banana or coral glow, same snake, maybe just different lines, but I think it's a little bit hard to tell unless someone is absolutely for sure that they have gotten this coral glow and only bred it to coral glow line before. And I'll do a video on line breeding at a different time. If that's confusing, don't worry about it, but check out that video when it comes out. So what's so different about bananas? Well, let's say you get a banana clutch. Almost all, if not all, of the males will be banana. And I have heard people say that it's like 60% all the way up to 90% of the offspring will be males that are bananas, and the rest of them will be females. Well, that's really strange, and you can imagine it boggled some people's mind for quite a long while until they begin to figure out the sex gametes. What sex gametes are the sexual cells that a mother and a father produces, like the sperm and the egg. And what do they contain? Some reptiles, it's very different than mammals, but ball pythons are the same as us. It's the X and the Y chromosomes that are passed on. So if you're XY, you're male, and if you're XX, you're female. So this gene is actually sex-linked. I mean, this gene occurs only for males on the Y of the X and the Y. If the banana gene can only occur on the Y, then that is why you don't get female bananas, because there's no Y cell in the female makeup, which is XX. And so what you have is all of these males being bananas and all the females not being banana. But something really rare and really strange can happen, which is called a crossover, and a crossover occurs when, let's just say, they have the Y and the X right there, the gene of the coral glow gene or the banana gene crosses over to the X, and now all the females are going to be banana. But it's not as simple as that. Alright, so let's just talk about this from like a breeding standpoint. So if you have a male banana and you breed it to another ball python, almost all of your offspring that are banana are going to be male. Now, the only reason this wouldn't happen, and if one banana was a female, is if that really super rare and awesome crossover happens, where the banana gene goes over onto the X chromosome instead of the Y chromosome. And what happens then is that you take that female banana that you got, you grow it up, and now that female banana, when you breed her, she's going to throw around that X chromosome like crazy, right? I mean, that's all she can give is an X, because that's all she has is the X chromosome in her. She's XX. And that chromosome has the coral glow and banana gene on it. So what ends up happening is her offspring aren't skewed anymore by the trait of banana or coral glow. Just like if you had a pinstripe ball python, the chances of getting another pinstripe aren't going to be, oh, if it's a male, it's going to be, this much of a chance, and it's a female, there's going to be this much of a chance that it's going to be pinstripe when the offspring is born. No. Now, every single offspring that she produces has the same chance of being banana or not. So that's pretty awesome, right? But let's just say you have a male banana in that clutch. And you take that male banana from that clutch, you grow it up, and you breed him now. What happens is that he's only going to produce, or mostly going to produce, female bananas. And why is that? Well, because when he throws his X chromosome, just like his mother's, it now has the banana/coral glow gene on it. How cool is that? So those are called female makers. And if you have a banana/coral glow male that creates banana/coral glow male offspring, that's obviously called a male maker. So the male makers, again, just going over this, the male makers have the banana/coral glow gene on the Y chromosome, and the banana/coral glow males that produce mostly female offspring are going to have the the banana/coral glow gene on the X chromosome. So that's pretty awesome. So let's just say we have a female maker. What's going to happen if he produces a male banana/coral glow after, of course, that crossover occurs? Well, that male now has the banana/coral glow gene back onto the Y chromosome. Basically, the terms to come away with this are female maker is a male banana/coral glow that has the gene for banana/coral glow on the X chromosome, and a male maker is a banana/coral glow that has the banana/coral glow gene on the Y chromosome. And also to know crossover is just when one sex-linked gene that's normally on, let's just say, one of the chromosomes, let's just say the Y chromosome, gets crossed over onto the X chromosome instead and passed on to one offspring that way. And so I hope that helped understanding the magical banana coral glow gene, because it did boggle minds for a long time, and now we're finally just starting to understand it. It's an incredible incomplete dominant trait. Now this next snake I'm going to show you is from Reptile Collective, and it is a paradox. This snake has been named Skittles by them, and it is pretty infamous. As you can see why. It is just amazing looking. There's all different kinds of each one of its genes put in there, and this is kind of what I would imagine codominant to look like, but again, this is just a paradox. It's nothing else. And so the genes that are in here are black pastel, super pastel, albino, pied, and then paradoxing. Now, paradoxing so far has not been determined to be genetic. Now, there are some people out there, and I've even talked to some, who believe that there are some lines that are genetic. There are some genes that cause more paradoxing. I've even heard tell of a new gene coming up that causes paradoxing, but so far I have not seen enough definitive data to support that, and so I think right now it's safe to say that paradoxes are just something really amazing that happens every once in a while. It's just kind of like a fluke in the genes that we can really appreciate. Now this girl came from an albino black pewter to a pied female, and he states in his for sale ad for this girl is for sale by the way, that those females have produced 5 paradoxes total, 4 of them with 1 pink eye and 1 black eye. So that can sort of lean to maybe something else going on here. Maybe this is genetic. Maybe this line right here has something more going on. So only time will tell. It's pretty exciting if that's true, because man, this snake, dang, I mean, that's pretty incredible to see that, to see this animal. It's just like such a mess, yet beautiful at the same time. I'm just really excited to see where it goes, and really good for them to trying to prove out something that most people have already put their foot down and said, no, this isn't going to ever be genetic, because it never has been genetic. I mean, you don't know until you try, right? So that is a paradox. So this is an example of a possible chimera. Now, they meaning the people who created this, who is Freedom Breeder, and who this photo is from, state that they think that this may be a chimera. Now, a chimera would be the joining of 2 embryos to create 1 animal. A good way to find out if this is really the case is if you take DNA from separate places on the animal. It should, and sometimes does, come back as 2 different animals, which is really amazing. They've named this guy Harvey Dent for obvious reasons. Half of him is scaleless and half of him is not. And he is so cool looking. I mean, holy cow. There's like nothing else out there that looks like this guy. I think there's been a few other snakes that have hatched after him that started looking sort of like him, with like half of their body scaleless and half their body not. So maybe the new scaleless has some sort of odd effect that this can happen, or maybe this really is a chimera and we're just getting really lucky. Now here's another picture where you can see it a little better and a lot clearer on the belly there. And again, this would not be something that is genetic. This is just something that happens in breeding and happens just randomly, and it's, again, just amazing just to sit back and look at. Now, not everything is fine and dandy in breeding. You really need to know the genetics of your snake and know what happens when you mix it with other genes, because there can be some really, really terrible potentials there, or some really, really amazing ones, as we've seen in this video. So if you'd like to see another video of possible fatal gene combinations or dangerous gene combinations, please let me know, and I would love to do that for you guys. So I hope you guys are doing awesome. I hope you guys are going to have some great holidays coming up here. Please leave a like if you think that this deserves it, and definitely share it if you think that it deserves that as well. I would love for other people to be able to use this video as a tool. And like I said before, the link is going to be in the description for 2 different kinds of genetic calculators. They don't show you the Punnett squares that we did, but now you should be able to make those on your own, hopefully. They do, however, tell you pretty definitely what the combinations are going to be, and sometimes they even have photos and references. So definitely fool around with those, take a look at it. I would recommend Morph Market just because I use Morph Market and am more familiar with it, so I can't really recommend World of Ball Pythons just because I don't use it. And let me know what else you'd like to see from us. I'd love to build this community around what we all want, and not so much what's easiest for me to do or what takes the shortest time. Also, the link to our website is down below as well. Give that a look if you have any questions. We have some informational Google Docs and also our availability. So thank you guys so much again for stopping by and watching this video. Really enjoy doing this. I really enjoy seeing the responses, and we'll definitely catch you next time.